You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have Dr. Kenneth Witwer. He's an associate professor of molecular and comparative pathobiology and neurology at Johns Hopkins University School of Medicine. His research focuses on extracellular vesicles, sometimes called exosomes, RNA-mediated regulation, biomarker discovery, and therapeutic modulation of innate and intrinsic defenses. So, Dr. Witwer, aka Ken, thanks for coming. How are you doing?
I'm doing fine, thanks. Glad to be here.
Yeah, if you don't mind, exosomes are a very exciting topic to me, or extracellular vesicles. I wanted to ask you some questions about that, if that's okay.
That sounds good.
Okay, well, what in particular, how is your research looking at EVs and their effects?
So I'm interested in extracellular vesicles, or EVs, largely because of my past. So when I began to do some research in science as a beginning graduate student, my interest was in HIV, the human immunodeficiency virus, and specifically how HIV and related viruses will infect host cells, and how they will also contribute to disease in the host organism. And so HIV, of course, it's a small particle. It's a particle that's released by some cells and then taken up by other cells. And it's a particle that is delimited by a biological membrane. So just like all of our cells, the virus itself, which is made by our cells and could be seen as even a part of the cell, something that is released by the cell, is going to contain this biological membrane that determines its ability to infect other cells. And in this sense, the HIV virion, that is the infectious particle of HIV, is itself an EV. It's an EV because it has the lipid bilayer membrane, so this membrane from the host cell, and it's an EV in that it contains materials from the host cell. It also contains materials that the virus is contributing. But I find that the extracellular vesicle, one of the best ways to describe how extracellular vesicles work is to invoke the example of an infectious virus, because the virus has a particular set of cells that it will infect, and it has specific effects that it exerts on the recipient cell. So that's what.
Right, so this is super interesting. When a virus first infects a cell, does it have a lipid bilayer, or is it different? I thought it just had like a protein coat, and the exosomes that it, I guess, forces the cell to put out, are they very different than the original virus that infected that cell?
Yeah, that's a good question. So, of course, we have a very wide range of viruses that can infect cells. Some of them, indeed, just have a protein coat. So we could speak of enveloped. Those are the viruses that would have that membrane, and then non-enveloped viruses. So the enveloped viruses, the way that they would get into a cell is by actually fusing. So the two membranes, so if you think of like two big soap bubbles that if you push them together, they might eventually fuse so that their membranes become one. And this is what happens with HIV. and with related enveloped viruses, they actually fuse their membranes with the recipient cell and then insert a payload, if you will, of infectious material that can then sort of take over the recipient cell. So you also asked about the EVs that might be produced by an infectious cell. And this too is an important question because once a cell is infected, it does to some extent the bidding of the virus that has infected it. So for HIV, for example, you would get production of the genomic RNA, so that's the genome of HIV, and you would also have production of proteins that are important for the HIV virus life cycle. So together, these proteins and RNAs would be packaged into virions, the infectious virus. But in addition to those virions, the cells can also make a broad range of extracellular vesicles that would contain everything from 0 components of the virus to many components of the virus, while still stopping short of having the full complement of viral components that would be necessary to infect another cell. So in that sense, you have a very broad range of particles that can be produced by the infected cell.
But wouldn't that mean that the cell is either intentionally or unintentionally broadcasting the fact that help? I've been attacked by a virus. Come kill me or come stop this thing. If it gave off EVs with incomplete, viral components that rendered them ineffective, wouldn't that be an alert again to the immune system of the host?
That's right. So you could have communication of this infected status using the EVs that that the cell would produce. Now, HIV has an interesting aspect to its life cycle in that it can become latent. So the latent virus, the way that we think about a cell that is latently infected is that, the genome is there, the viral genome is there, but it's not doing very much. So it's not producing these proteins that are necessary for, generating an infectious virus. Now, why would a virus want to do that? Because you would think that there would be an urge to, or a tendency to replicate. And in fact, that is what a lot of viruses do. But HIV and I think related viruses are really going for the long game, where they would like to infect cells and ultimately become part of the host. So if a retrovirus can get itself into the germline of the host, then it has the opportunity to be in every cell of the progeny, that is to say, the next generation of that organism. And so even though it might not now be replicating anymore, it will persist. It will persist for generations. And in fact, if you look at our genomes, you will find that there are many, many retro elements. Some of them coming from ancient retroviruses, retroviruses like HIV, I should say, that are present in the genome and that actually constitute a very large percentage of our genetic material.
Yeah, I've heard 7% approximately of our genetic material is endogenized retroviruses.
Yeah, and so these retroviruses, what are they doing now? So you can find some organisms where you do have endogenous retroviruses, we call them. These are the retroviruses that are transmitted through the germline that can have the potential for replication. We don't think that the human retroviruses can do this now, the endogenous retroviruses. However, we do know that there can be expression of some of these viral components, so viral proteins. And it may even be that in certain physiologic conditions, there would be expression of some of these viral proteins that could be displayed or anchored in the membrane, the lipid bilayer, that could actually potentiate the roles of extracellular vesicles in these conditions. So conditions like pregnancy, for example, or certain cancers, there could be more expression of proteins that would enable extracellular vesicles to communicate between cells.
But if a virus becomes part of our DNA, can it be reactivated at a later time? Let's say it happens to me and I survive the virus and I have offspring and it's gone into my germline and my offspring have it now as part of their DNA. Are there circumstances under which all of a sudden my offspring cells could make more of those viruses in an active form or in some kind of form that would perform some function?
So that's probably possible. It's theoretically possible if you had some recombination events where you were able to get a full complement of genes that were necessary, viral genes that were necessary for this replication. Then you could potentially have a reawakening of an old virus. Or another possibility would be an infection with a new virus that might complement some of the activities of the older viruses. So these are things that are that are theoretically possible, but we don't have examples of them happening in humans to date.
But do we have examples of, again, an endogenized retrovirus now changing my phenotype or my offspring's phenotype, changing how we, like you said, the membranes of my cells allow certain things in or out, or other bodily functions? Like what function can these endogenized retroviruses take on in the progeny of the person infected?
Well, I think that you could have some contributions from viruses at both the RNA level and the protein level. At the RNA level, of course, we have functions of RNAs that will contribute to transcriptional regulation. Also, at the post-transcriptional level, we could have small RNAs, for example, that would regulate translational events. At the protein level, we could have proteins that can encourage fusion, so that membrane fusion that I mentioned. We may be able to shift the balance away from a more restrictive environment for EV-mediated communication to one that is more permissive for that communication. And why would that be important? Well, it could be important to facilitate cell-to-cell communication, to deliver functional molecules from one cell to another. And these could be things that are beneficial to the host, or it could be that they are detrimental. For example, in the case of certain cancers that may want to communicate messages to each other or to non-cancerous cells, that could be more beneficial to the cancer than to the host.
I know I've taken you down the virus path, but I just have one more question there. Has science seen evidence of epigenetic marks that happen to regions where, regions of endogenized retroviruses in our DNA? Because if that happened, maybe that would signal that it has some function in the body that need up or down regulate.
Well, that's a really interesting question, and I don't study endogenous retroviruses at that epigenetic level, but yes, I think that what you're alluding to here would be the down regulation or the chromatin. Let's give an example of chromatin modification that would keep endogenous retroviruses silenced. In most physiologic conditions, that might indicate then that you want to keep a tight rein on what those endogenous retroviruses are able to do physiologically. So I think that's a very interesting question, and one that I think probably merits more investigation in different physiologic states.
Okay, yeah, interesting. Has anyone been able to profile? I don't know. I mean, I know EVs are really tiny. Has anyone been able to profile the extent of the EVs that are put out by any given cell? Are there dozens or hundreds of kinds? And how does the cell interpret the incoming EVs? And how do they know where it's coming from, what the intent is, what it contains? And it just seems like a whole huge effort to not only, again, put them out with the right targeting and the right packaging, but also bring them in and interpret them and utilize them.
Oh yeah. So those are all great questions, and it's a very wide variety of questions there. So I'll try to answer here. To start with, I think that historically in the field there have been two main categories of vesicles that have been investigated. So if you go back to the 30s, 40s, 50s of the last century, when electron microscopy really came into the forefront and people were able to look at tissues and at particles that were coming from biofluids, and they were able to say, wow, this is what a virus looks like. And here's this other thing. I mean, or maybe it's the same thing. Maybe it's a virus. Maybe it's something else. They were able to develop the concept of the extracellular vesicle by looking at these electron microscope, micrographs. And it wasn't really until maybe the 1970s or so that there was an appreciation that these vesicles could be formed by budding of the plasma membrane. So that's the membrane that surrounds the cell. And this was actually investigated in bacteria, in other single-celled organisms, and then also in mammalian cells and other mammalian samples like biofluids. So this idea of budding from the surface was one form of generating an extracellular vesicle. The other one that came a little bit later was the idea of budding into an internal compartment of the cell. So the endosomal compartment of the cell is an extensively studied subcellular set of compartments. And one of these types of endosomes is called the multivesicular body. It's defined by these particles that are inside of it that have budded into it, that when they are released from the cell have canonically been called exosomes. And so these tend to be slightly smaller than the particles that are released from the plasma membrane, simply because they can't be larger than the multivesicular body itself. So these two, let's call them biogenesis pathways, have been most extensively investigated. But what I find really fascinating about the field right now is that we're finding that there are many types of vesicles that we did not appreciate before. One type is called the migrosome. So this would be like an extracellular vesicle that's left behind by a cell that's crawling across a matrix. Another one is the large oncosome. And so some cancer cells will actually make very large partitions of their cytoplasm that they then release. These can be as large as some other cells, and they're defined by not having the presence of a functional nucleus. So these are very large bodies. So we now know that there are EVs that range in size from about 35-40 nanometers. That's about as small as you can make a lipid bilayer with some proteins inserted into it, all the way up to 10 microns across, in diameter or more. So there's a whole zoo out there that I think we were just around the entrance, I think, for many, many years, and now we're understanding that there's a lot more in the interior there that we can learn about. So the diversity of extracellular vesicles is certainly a hot topic right now. You asked about the recognition of extracellular vesicles by cells, so by recipient cells, and this is another area that I think is very important for the field, because we would like to develop extracellular vesicles as therapies. And if we're going to be treating a condition with EVs, we have to know how many of our EVs are going to go to the cell or the tissue of interest. So deciphering the surface components of EVs is the key thing that we have to do to be able to deliver them. So the idea here is kind of like a lock and a key, right? So on the recipient cell, you might have a few different proteins that are displayed, and on your extracellular vesicle, you want to have complementary proteins that are displayed. So proteins that will be ligands, interaction partners of these cellular proteins. And I think a great example of how this works actually comes again from the virus field. So with human immunodeficiency virus, HIV, there is a set of proteins that's on the surface of the virion that are encoded by HIV. And so these proteins need to find binding partners on a recipient cell. So one of these binding partners would be the CD4 receptor. This is a receptor that's found on human T lymphocytes and then also on human monocyte lineage cells. But there can't just be that one receptor. You also have to have a partner on the recipient cell. And there are several of these that have been documented in HIV research. So when the virus, its proteins, can interact with those partner proteins on the recipient cell, only then will you have a close interaction of the virus with the cell to the point that you can achieve membrane fusion. So a big part of what we as EV researchers are trying to do today is to identify those combinations that allow EVs to communicate with the recipient cell and potentially be taken up by that cell.
What about an experiment whereby, let's say, you culture liver cells and harvest the exosomes that they produce, and then put a whole bunch of those exosomes into a culture of, I don't know, pancreas cells, and see if that induces the pancreas cells, let's say, uptake them, and see if that induces them to produce different exosomes than they were producing before.
Yeah, so that's one way to approach that question. You could use libraries of EVs that you have produced from different cell types, where you know the surface composition very well, and then put them onto to recipient cells that might very well have different combinations of surface receptors. Another way that we can investigate this is by doing some sort of a screening approach that involves genetic modification. So we could take a particular cell type, modify its membrane protein. So we could perhaps go into its genome and selectively delete certain membrane proteins, and then observe whether a particular EV type could still exert an effect, a known effect on that cell type. And by going through our entire library of different membrane genes, membrane proteins, we could identify those that are involved in the EV interaction, right? So we could do this at either the level of the producing cell to modify the surface of EVs, or we could do this at the level of the recipient cell to modify its surface proteins to understand these two poles really of the interaction.
Well, do we understand, let's say, a cell that takes in EVs, that they have to fuse with the cell's outer membrane, but then where do they go? Do they go to certain spots within the cell and then become active? Can we even observe that?
Yes. So what we know is that when an EV is taken up by a cell, typically what will occur is not so much a surface fusion, but rather an intake into the endosomal compartment. And then if the EV is to deliver its cargo into the cytoplasm of the cell, it must fuse with that endosomal membrane. And so this is also a key thing for drug delivery. So we want to have what's known as endosomal escape. So getting out of that endosome before the endosome ultimately destroys its contents for recycling. So we know what's happening with certain viruses in the endosomal compartment, and that you need some degree of acidification to achieve fusion with the endosomal membrane. But I think that we're still working out what needs to happen, what the prerequisites are for the fusion of an EV with the endosomal membrane. And it's interesting if you look at some of the literature that's out there on EVs and where they go inside the cell. You can find some very nice images of fluorescent labels. So in many cases, you want to label your EV population somehow. One way to do that is to label with a membrane dye, and this membrane dye would just insert into the lipid bilayer, and you can then deliver the EVs to the cell and see where that label goes. And so there have been some very intriguing images of these dyes that are in endosomal compartments, maybe even in a perinuclear region, that seem to persist for hours or even days after delivery of the EVs. And so it's a bit of a mystery as to what exactly they're doing there. Does this mean that they are not functioning? Could it be that we're just looking at the dye and the EV has gone and its cargo have gone somewhere else? So that's a mystery, I think, right now in the field, and we're trying to figure out what is happening there. But I want to bring up something else, which is that the delivery of cargo, like we would see with a virus, may not be necessary for some of the functions of EVs. And in fact, it could be that simple interaction of the EV with the surface, with a surface protein, for example, of a cell, would be sufficient to start the cascade of effects that we know are occasioned by EVs. So it could be that the EV is more of a signaling platform than a delivery vehicle in many of the effects that have been characterized for EVs.
What about, where are some of the places that EVs are made within a cell? You said maybe the outer membrane will butt off, but Are there other, I mean, I don't know, does like the endoplasmic reticulum make them and package them? Like, where do they come from within a cell?
Well, the classical endosomal budding, so the inward budding into the endosome, I think is probably the most studied. But certainly, I mean, I think that we know now that there are some EV populations that will have that will contain endosomal markers, Golgi markers, nuclear markers. Just because we found some of those deeper cellular markers does not necessarily mean that our results are wrong. It could be that there very well are contributions of these compartments to EV contents. So whether the membranes from these different compartments are actually giving rise to EVs, or whether there is just communication of some of those, or I should say conveyance of some of those contents into the EVs, I think remains to be seen.
And I don't know if this is accurate or not. I don't know how much the various constituents of, like, let's say the human body communicate with each other. But, let's say I'm a liver cell. I'm making my own EVs, whatever, but I'm also, again, taking in EVs from other liver cells, other organs, maybe outside, viruses, et cetera. Like I would think I would have to have an interpretation system that would help me differentiate, okay, this is, one of my own, another liver cell that has sent me an EV, or this is maybe from the pancreas, or this is from the brain, or this is from, a virus, and interpret and know what to do with these things somehow.
Yeah, so you have all these different messages that you're being bathed in, right, as a cell. Right. And so you have to be able to sort those messages, because they can have very, very different results. So I believe that one function of EVs is simply as nutrition, or we could call it molecular recycling. So there might be certain EVs that are safe for the cell to take up and to recycle. So you don't have to make all of your own components. You might come across some nice packets of molecules that you can reuse, and that might be one big function of EVs. Some EVs might convey danger signals, right? So they might provoke inflammation, and so you might want to be careful about taking up too many of these. And then there are other EVs that are genuinely conveying messages that you need and that you want to be able to go about your daily business. And those vesicles then are ones that you really want to pay attention to. So it's hypothesized that the different vesicle populations are going to have different signatures on their surface that will distinguish between these different end uses.
Yeah, it's like, it seems just like a language you'd have to know what to produce, how to produce it, what to take in and interpret, how to interpret it, what to do about it. Where do you think this orchestration comes from?
So I think that it really does come down to the combinations of molecules, proteins for sure, but possibly others, that can be displayed on the surface of EVs. And this does lead us to a very big challenge in the EV field right now, and that is to be able to do single vesicle profiling, phenotyping we call it, deciphering what those combinations are on the EV surface. And this is actually not a trivial thing. So if we look back to the 1970s, where immunology was back then, there had been a technology that was developed and was first being used in the 1970s to understand immunology. And this was called flow cytometry, right? So this is a technology where you could look at different proteins that were on the surface of a cell and try to understand which molecules were on different cells. Today, some of us are using a modification of flow cytometry to look at small particles. We're using various technologies actually to try to do this single particle profiling. And the idea here is to maybe take 4 or 5 markers, or 2 or 3, and understand what combinations of those are on different vesicle populations by examining vesicles at the single particle level. So I think that we've made a lot of progress just in the last 5 years in being able to do this. And as we move forward, and as this technology becomes more sensitive, we're going to really be able to understand more about the true diversity of the extracellular vesicle population. Much like immunology has developed over the last few decades as a result, that pioneering work with flow cytometry, where now we understand that there are many different subsets of our immune cells that previously we could not define. But now, because we're able to look at the single cell level, we know that these different types exist. And so, the challenge is just the physics. It's the sizes that we're talking about. So if a cell is if we look at what we can see in terms of a single cell, the average extracellular vesicle might be just a millionth of the volume of that cell, or even smaller. So we need very sensitive techniques to be able to achieve this goal of single vesicle characterization.
In terms of EV therapy, are there any therapies in use right now, or ones that are close to, going to the clinical trial level?
Well, interestingly, there was an EV therapy that was introduced back in the 1980s. So this was a vaccine, actually. So a vaccine that was made from outer membrane vesicles of bacteria. And as we look back at this history, there have actually been multiple vaccines now that have come out that have been made from bacterial EVs. We can also think about what is necessary to create other forms of therapy. So let's say we're going to make a cancer therapy, or we're going to deliver a particular molecule to cancer cells and maybe convince them to give up. How do we get enough material to be able to do this? Well, we have to make these EVs from the patient's cells, or can we make them from some cell line? Or even better, could we use something like a bacterial system? Some people are using vesicles from different natural sources, like different foods. Can we mass produce vesicles, in the quantities that are needed to have therapies that can be given to multiple individuals? So it's always fun for me to look back at this history of EV therapy and how it can inform what we're moving towards in the future.
I guess by studying how EVs would end up entering a cell, you can learn a lot of things. You can learn how to package things to get into the cell better. And to interesting. So what's I don't know, any particulars right now in the research that are anything like super interesting that you're finding? Not that everything we talked about so far isn't super interesting. Anything in particular you're seeing that's really piquing your curiosity?
I think one thing that we that we haven't touched on yet is how we can use vesicles in diagnosis and prognosis in medicine. So this concept of a surface profile, this lock and key kind of concept. It's not just about delivery, it's not just about therapeutics, but it's also about tracing the vesicle back to its origin. So in the blood, there's I think you can find a lot of interest in the news about blood tests that people have developed for different cancers, for Alzheimer's disease, and so on. But is there some way that we could develop a very broad screening test for different conditions, different disorders, using extracellular vesicles? And I think the answer to that is yes. I think that we have a lot of opportunities now to use extracellular vesicles to understand the health of a particular cell type or a particular tissue. So to give an example here, my lab does a lot of work on central nervous system diseases, neurodegenerative diseases, starting with the HIV-associated neurocognitive disorders. And we find that other neurodegenerative diseases like Alzheimer's, Parkinson's disease have some commonalities. Is there some way that we could identify a population of extracellular vesicles that have come from the brain that will tell us about the health of neurons and other cells in the brain that are affected by these diseases? I think what we need to be able to do that are markers that are specific for the cells of interest. And so to some extent, we know some of these markers today. And then methods that will allow us to capture the extracellular vesicles that display those signatures from, let's say, a blood sample or a saliva sample. and then allow us to really assess what's going on in the affected tissue. So this is an area where I see a lot of work going on right now. We see a lot of different technological platforms where engineers, chemists are working together with the biologists to try to really push the envelope of what's possible. And I expect that there will be some very interesting developments here in the next few years.
Has anyone tried to characterize what the outer membrane of all the cell types, like in the human body, look like? What proteins they have, what surface structures?
Yeah, so I think there's a wealth of proteomics information that's out there that we can use to help inform our biomarker searches. So certainly there's a lot of public data, and I think that there's an opportunity to gather even more so that we can look at specific compartments of the cell that might be releasing these EVs.
Right, but the outer membrane itself of all the cell types in the body, I just wonder if anyone's characterized them and looked at the differences, seen how similar and how different they are, or is that just a massive undertaking to do something like that?
Well, I would say that I think there's still a lot of opportunities to do this for cell subtypes. For the very broad for the very broad cell types, I think that we already have a lot of information about those membrane, transmembrane proteins that we can follow up on in our EV research. But for subtypes of cells, I think there's still a lot of data that can be gathered. So that's a great idea for a research project to pursue. So if we could really map every cell in the body and what its membrane complement would be in health and disease, that would be very useful.
Even if they did it for one particular organ, the brain or the heart, or let's say the heart, let's say it has 40 cell types. You can map all of them, and I would think you'd have a much greater understanding of how things could affect the heart and all the constituent cells of it.
Yes, yeah. So I think if you if you look at brain, the brain research, you look at the cancer atlases, you could do this for really any organ in the body.
It's kind of it's a question, in a very different direction. But do you think there's EV communication between our microbiome and our somatic cells?
Yeah, that's an area of interest as well. Where you have many organisms that are living in the gastrointestinal tract, for example, and that we know are producing EVs. So I think every organism that we've looked at so far is making EVs of some sort or another. Do they communicate then with the host cells? There's some evidence of that. There's some evidence of some transfer of biologically active molecules. But I think the jury is still out as to the extent to which that communication is happening, and then also the effects of the communication. And then also, does the communication go in the other direction too? So could, for example, gut epithelial cells be releasing EVs that would influence the growth, the behavior of organisms in the microbiome? So there's, of course, with all the diversity in the microbiome, I think this is I think that there are a lot of questions to ask, and it's sometimes difficult to answer those questions by choosing the right simplified system to use.
Yeah, it's crazy if you think about the environment that a skin cell sees. And then you look at like a gut epithelial cell, and then you look at maybe something that doesn't see very much, the interior of the brain, or all these three different types of cells have a very different environment about them, and different actors and characters, molecules, and it's just crazy that they could all function and somehow interpret what's going on outside of them, and keep their homeostasis and keep themselves going.
Yeah, if you think about the networks there, all the different connections between these different species, and where you have, probably not only EV-mediated communication, but also other signaling molecules. There have been some fascinating studies that have been done in plants, for example. So you have plants that can be colonized by bacteria, by certain fungi, And you see that this EV-mediated communication is happening in different directions there. So there's just a wealth of opportunities now to understand how EVs are contributing to the environmental interactions of different species. I think it's a really fascinating time to be doing research in this area.
Yeah. Well, what do you think is ahead for you in the next few years? Any big discoveries that you feel like you're getting close to making?
Well, I think one of the things that we would like to do is to come up with better delivery methods for extracellular vesicles to get them into the brain. So this is one thing that we are working on, and I know many others are working on that too. So how do we get a therapeutic vesicle across the blood-brain barrier? That's one thing that I would like to get some good answers to in the next few years. And another thing I think is the biomarker angle. So what is going to be physically possible for us as far as distinguishing different subsets of EVs by their origin, by their cellular origin, when using a blood sample? How much blood are we going to need to be able to achieve a good diagnostic test for a particular condition? So there are, obviously many groups that are working on this. I think we've had some successes in certain types of cancers, and I would like to see that extended into other diseases as well. And then I think for my lab in particular, we would like to come up with ways of either suppressing HIV production or stimulating HIV production so that the immune system can kill off the HIV-infected cells using extracellular vesicles as a means of delivering certain molecules. So I guess these are the areas that I hope to contribute to in the next few years.
What would be interesting is if you looked at the EV communication surrounding apoptosis, and you could stimulate that in a population of cells with the right EVs. Those cells must be giving off something, letting the surrounding tissues they're going to do that. But first, the surrounding tissues must be telling the cell somehow, probably with EVs, hey, it's time to commit Harry Carey there.
Yeah. So, I think that whether you're, making sure that a cell is recognized by the immune system and eliminated, or convincing the cell to eliminate itself. Either of those would be a satisfactory outcome. When you have a cell that is creating problems in its microenvironment.
Yeah. Well, very good. I appreciate your patience and all these crazy questions. What's the best way for listeners to find out more? Maybe get in touch with the lab or find papers that they're interested in?
Yeah, so I would just like to refer the listeners to the International Society for Extracellular Vesicles. So if you look up isev.org, that's isev.org, you can find a lot of information about EVs, including links to 2 massive online open courses. So we have a very basic course, and then we have one that's more focused on different functions of EVs in health and disease. And so these have been widely subscribed to. A lot of people have taken them, and they're free to take. And then there's also a journal that's devoted to the field, the Journal of Extracellular Vesicles, or JEV, that of course publishes scientific papers about extracellular vesicles. Also on the isev.org website, you'll find a link to an animation that's been made that's kind of an introduction to the field. You can watch that on YouTube, and there's also a high definition version that you can watch in kind of virtual reality. So this website, I think, will give you a lot of information about EVs at both the introductory level and at deeper levels.
Excellent. Okay. Well, very good. Well, Ken, thank you for coming. It's been a great call. I appreciate it.
Great to talk with you. Thanks so much.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.